Do you believe in evolution?
I believe in I believe in microevolution. I believe that there are real evolutionary processes. I'm skeptical about what's called universal common descent, the idea that all living forms have evolved from one single common ancestor. I'm profoundly skeptical about chemical evolution, the idea that the non-living chemicals in a prebiotic ocean or prebiotic soup arrange themselves to form the first living cell. And I'm also skeptical about the creative power of the mutation selection mechanism, which, as it happens, so are many leading evolutionary biologists today. I attended a conference in 2016 at convened by the Royal Society in London, Royal Society being the oldest and most august scientific body in the world. And it was convened by a group of evolutionary biologists who were essentially dissatisfied with neo-Darwinism, the standard textbook theory that we learn in all high school and college textbooks. And many of them were saying we need a new theory of evolution. The first talk at that conference was given by Gerd Müller, a prominent Austrian evolutionary biologist, and he simply enumerated the five major what he called explanatory deficits of neo-Darwinism. And his basic perspective was the mutation selection mechanism does a good job of optimizing or modifying pre-existing forms. It can generate small-scale variation, but it does a very poor job of explaining the origin of those forms. Think about, for example, Darwin's finch beaks. Great job of explaining how variations in weather patterns result in changes in the shape and structure of the finch beaks. But that mechanism turns out not to do a good job of explaining the origin of birds or other major animal groups in the first place. So modification, yes. Innovation, no.
But modification over massive amounts of time, don't you think that would eventually lead to new groups? Because a lot of new groups have similar origins, or at least origins from one ancestor.
Well,
like primates.
Time was always the hero of the plot. But let me just run a couple of arguments by you, and let's see what you think. Okay, and I developed these in a lot of detail in my book Darwin's Doubt. If we now know, thanks to the genetic revolution, the molecular biological revolution, that if you want to build a new form of life, you have to have new code, because all new forms of life depend upon new anatomical, a fundamentally new type of animal, for example. So you need new anatomical structures but the new anatomical structures require new cell types, new types. So if you got animals that first come on the line and they have a digestive system, they have a gut. Well, you got to have enzymes that can service a gut, that can process food. So enzymes are types of proteins. Proteins are built from the informational code in DNA. So anytime you want to get it's just like in the computer world. If you want to give your computer a new function, you've got to provide new code. So we have these long digital bit strings of A's, C's, G's, and T's, not zeros and ones, but A's, C's, G's, and T's in a digital string, and we call that a gene. And if you have a section of DNA for building a protein, that's great, all works. Now, but if you want to build a fundamentally new form of life, you got to have new proteins to service the new cell types to build the new anatomical structures. In our computer world, we know that if you start randomly changing the zeros and ones in a section of digital code, you're going to degrade the function of that code long before you come up with a new string for making a new program or operating system. The functional sequences they're highly isolated in what's called sequence space. You can change a few things and still retain function, but after a very few number of changes, you're going to degrade the function, and long before you come up with a new function. Now, the Darwinian mechanism starts with the idea that there are random changes in those digital bit strings, those sequences of A's, C's, G's, and T's. And based on our experience in the computer world, we would expect that random changes are going to, again, degrade those strings long before they're capable of building a new protein. And there's now very compelling experimental evidence that that's true. There's an Israeli molecular biologist, Dan Toffick. Unfortunately, he died fairly recently in a tragic accident. But he was doing mutagenesis experiments on sequences of code for building specific proteins that fold it into stable structures. They're actually called protein folds. And he found that between 3 and 15 mutations was enough to degrade the thermodynamic stability of the protein structure that the gene was making. And once you lose that thermodynamic stability, you have no functional possibilities.
Is there possibly an undiscovered mechanism for protecting against that we're not aware of yet?
Possibly, but there's numerous lines of evidence suggesting that that mutations are within limits. You can modify again. You can optimize an existing protein structure called a fold. But if you allow too many of those mutations, you're going to degrade. And long before you would get a fundamentally new protein structure, another protein fold. So that's just one of many. I want to run one other argument by you that I think is very intuitive. If you want to build, it turns out that there are structures or systems for building that are very important for building new animal body plans, and they're called developmental gene regulatory networks. They were discovered at Caltech by Eric Davidson and colleagues. Eric Davidson has also unfortunately recently passed away in the last few years. But what they discovered is that you not only have genes for building proteins, you have genes that are building for constructing molecules that send signals that tell the genome when to express other parts of itself. So you've got there's signaling molecules that are telling the genome when to turn this part or that part on in order to build the right proteins at the right time as new cells are going through cell division in the process of animal development. So if you go from 1 cell to 2 to 4 to 8 to 16, et cetera, you've got to, and as you have a developing animal form, there are points in that trajectory where it's important to differentiate one type of cell from another, and for certain types of cells, muscle cells as opposed to nerve cells or bone cells, to start to be constructed. And all of this is closely choreographed by these signaling molecules. So you get DNA that builds regulatory RNA that turns on another part of the DNA that then turns on that builds a protein for servicing a particular type of cell at the right time and not at another time.